You're watching BBC Learning Zone, helping you to get on with some GCSE revision now. And it's science today, as we focus on biology for the next 2 hours.
Hello and welcome to GCSE Bitesize. This programme will help you with your GCSE Science Biology exams. There are other programmes about chemistry and physics, and they'll all help towards a grade C in the Double Science Award. There's also a higher tier science programme for those who want to go further. The best way to revise with Bitesize is to work through the video in bite-sized chunks, taking one topic at a time. Each programme is full of useful information and sample questions for you to try. And there's more in the GCSE Bitesize books and on the GCSE Bitesize website at www.bbc.co.uk/revision. This Foundation Biology programme is in 5 parts: cell activity, humans as organisms, green plants as organisms, variation, inheritance and evolution, and living things in their environment. You can use the timings on the menu and the clock in the corner to go straight to the topic you want to study. The first section is about cell activity. This section on cell activity covers animal and plant cells, movement in and out of cells, the cell nucleus, and cell division. First, let's compare animal and plant cells. All plant and animal life is composed of cells. They're the basic building blocks of life. The next clip looks at a variety of human and plant cells.
All living things are made up of cells, countless millions of them, and there are many different kinds. For instance, there are skin cells, muscle cells, bone cells, blood cells, fat cells, nerve cells. Plants, too, are made up of many different types of cells. For instance, there's the guard cells around the stoma in a leaf, the cells at the tips of growing roots, the cells in the stalk.
But what are the key differences between animal and plant cells? The next clip explains.
All animal cells are surrounded by a thin membrane. Inside the membrane is the cytoplasm, and in the cytoplasm are a number of distinct structures. Nearly all animal cells have a nucleus. Plants, too, are made up of complex cells with many different organelles. The functioning of these organelles is literally the basis of life on Earth. Plants use sunlight to make food in a process called photosynthesis. Photosynthesis is not one simple reaction, but a whole collection of complex, interdependent biochemical processes. Most of these reactions take place in an organelle unique to plant cells, chloroplasts. There are hundreds of other biochemical processes going on in cells, and they all seem to work together smoothly and efficiently. So there must be some system of control of the biochemistry. And that's where the nucleus comes in.
For your exam, you need to know the features common to all cells and the difference between animal and plant cells. Set up 2 columns headed animal cells and plant cells and note down their similarities and differences as you watch the next section. Here's a diagram of an animal cell and a plant cell. Both cells have the same basic features. They both have a cell membrane, which is a very thin, partially permeable outer layer of proteins and fats. It holds the cell together and controls what goes in and out of it. Cytoplasm is the contents of the cell, where many of the chemical processes take place. The nucleus contains chromosomes, which carry genes on them, which act as a set of instructions for the cell. Plant cells have some extra features. They have a cell wall outside the cell membrane for support and strength, permanent vacuoles containing cell sap, which exert pressure on the cell wall to keep the cells firm and help the plant keep its shape, and chloroplasts, which contain chlorophyll for photosynthesis. If you need more time to complete your table, stop the tape and look back over this section. How did you get on? Your table should look like this. Animal and plant cells all have a cell membrane, cytoplasm, and a nucleus. Plant cells also have cell walls, chloroplasts, and permanent vacuoles. Don't confuse the cell membrane with the cell wall. All cells have a cell membrane. It holds the cell together and controls the movement of substances in and out of the cell. Only plant cells have a cell wall as well, to support and protect the cell. That's the end of the section on animal and plant cells. This section looks at the movement of substances in and out of cells. Diffusion and osmosis are two processes by which substances and fluids move in and out of cells through the cell membrane. Diffusion is the random movement of molecules of a substance. The net movement of molecules is from a higher concentration to a lower concentration. Osmosis is the diffusion of water molecules. During osmosis, water molecules move randomly across a partially permeable membrane. The net flow of water is from a weak solution to a strong solution. Remember, osmosis only applies to the movement of water molecules. But how does this work with cells? A large part of a cell and its surroundings is water, and molecules of water and dissolved substances pass in and out of the cell through the cell membrane all the time. The cell membrane is partially permeable. It allows some substances, shown in blue, to pass through, but not others, shown in red. Substances are transported across the cell membrane by diffusion, and water is transported across the cell membrane by osmosis. There's more about transport through the cell membrane in the higher tier program. There are many examples of diffusion and osmosis in the human body. For example, diffusion moves small molecules of digested food from the ileum, the small intestine, into the bloodstream. Diffusion also moves oxygen from the air sacs in the lungs to the capillaries, and moves carbon dioxide from the capillaries to the air sacs. Osmosis absorbs water from undigested food in the large intestine. Osmosis also absorbs water into the root cells and root hair cells of plants, and is involved in osmoregulation in the kidney. Here's a sample question about osmosis in plant cells. The diagram shows two cubes of potato, A and B. Cube A is in a concentrated sugar solution, and cube B is in distilled water. The cells in the potato cubes naturally contain a dilute solution of cell sap. After 1 hour, which is likely to be true for cube A and cube B? Do they gain more water than they lose? Do they gain and lose equal amounts? Or do they lose more water than they gain? Okay, this question is about what happens to the water in the potato. So it's about osmosis. And during osmosis, we know that water moves from a weaker solution to a stronger solution. Looking at potato A, the concentrated sugar solution in the beaker contains a lesser proportion of water than the dilute cell sap in the potato. So water will move from the sap into the sugar solution to try and balance things out. That means the potato cube A will lose more water than it gains. With potato B, the distilled water has a greater proportion of water than the dilute cell sap in the potato. So the water will move from the water into the sap to try and balance things out. This means that potato B will gain more water than it loses. That's the end of the section on the movement of material in and out of cells. This section is about the cell nucleus. What's inside the cell nucleus, and what does it do? As you watch the next clips, note down what goes on in the nucleus.
The basic building block for all life is the cell. The tools it needs to carry out its work are inside. These organelles vary, depending on whether the cell forms part of a leaf in a plant, the skin of an animal, or the brain of a human being. But amazingly, virtually every cell contains the recipe for the entire body. The recipe is spelled out in a chemical code. Each line in the recipe is a gene, describing a particular characteristic. The chemical code is in the cell's nucleus on long curled-up molecules called chromosomes.
Each cell contains a nucleus, which holds 23 pairs of chromosomes. All the information which is needed to make a human being is contained within these. They're made up of 2 strands of DNA twisted into this spiral shape. And along these spiral strands are found the genes, our own personal genetic codes.
You should have spotted that. The nucleus is the control center that contains the genetic information for the cell on chromosomes. Chromosomes are long, thin threads made of DNA and proteins. And a gene is a piece of DNA with instructions to make a unique protein. All the cells in the body have an exact copy of all the genes, and each individual inherits a unique combination of genes, unless they're identical twins. DNA in the nucleus is isolated from the rest of the cell contents and their reactions, so it's protected from the possibility of damage. Normally, the chromosomes in the nucleus are too thin to be seen, but they become visible when the cell is about to divide, when they shorten and thicken and appear as distinct threads. That's the end of the section about the cell nucleus. This section is about cell division. There are 2 kinds of cell division, mitosis and meiosis. First, mitosis. Mitosis is how all cells divide for growth or repair. The next clip explains what happens.
This is mitosis, the natural process by which cells copy themselves to grow new tissue, seen through a microscope and speeded up 2,000 times. Look closer still at a single cell, and it's clear mitosis is a very organized process. The action centres on the cell's nucleus, where the DNA is stored as strands called chromosomes. The chromosomes curl up and tuck themselves away once their job is done. A lot goes on when a cell divides, so it's easier to watch mitosis in a computer simulation than trying to make sense of microscope images. Here are the chromosomes made from DNA. Before a cell divides, the chromosomes copy themselves, duplicating the DNA. Then, as the chromosomes move towards the middle of the nucleus, from the ends of the cell, strands of protein reach inward. The membrane around the nucleus breaks down, the chromosome pairs line up at the center, and the protein fibers attach to them. Suddenly the fibers contract, and the chromosome pairs are split. Two exact copies of the original DNA line up at either end of the cell. Finally, the cell membrane splits, and mitosis has created two identical cells from one original.
The other kind of cell division is meiosis. Meiosis only occurs in cells which give rise to gametes, the cells that take part in sexual reproduction.
It all starts when an egg from the baby's mother fuses with sperm from its father. Both sperm and eggs are very special cells because each holds just half the genetic recipe needed to make a person. Because they're special cells, sperm and eggs are made by a special process. It's called meiosis. First, the DNA is copied so that each chromosome becomes two chromatids. The double chromatids line up 2 by 2, one pair from the mother and one pair from the father. Sometimes pieces of DNA cross over between them, making the new DNA unique. Then, rather like mitosis, protein fibers reach in from the ends of the cell as the membrane round the nucleus breaks down. The fibers pull the chromatid pairs apart, and new membranes form around the two cells this creates. These two new cells each contain only half the genetic recipe for a whole person. Meiosis isn't complete until the new cells divide yet again. Once more, a fiber spindle forms to split the chromatids apart. In a man, they all develop into sperm. In a woman, one of them will become an egg. Meiosis only happens to make these special sex cells special because they hold just half the genetic recipe for a whole person.
It's important to know the differences between mitosis and meiosis. Draw up 2 columns and have a go at jotting down the differences. If you want to, go back and have another look at the 2 clips. To start with, mitosis cell division happens to all cells, while meiosis only happens to sex cells. In mitosis, the cell divides once to create 2 new daughter cells, which are both exact replicas of the mother cell and have the same genetic information. In meiosis, the cell divides twice to create 4 new daughter cells, which are not exact replicas of the mother cell and have only half the genetic information. There's more about cell division in the higher tier program. That's the end of the section on cell activity. This section is about humans as organisms. In this section, we'll cover nutrition, blood circulation, breathing and respiration, the nervous system, hormones, homeostasis, and human health. First, nutrition. This section on human nutrition is divided into digestion and the role of enzymes. First, digestion. Digestion is the process by which food is broken down into simpler substances, which can then be absorbed into the bloodstream. Energy in the food can then be used by the body. You need to know what happens in each part of the digestive system, so it's useful to summarise everything in a table like this. In the left-hand column are various organs of the body involved in digestion. Your task is to fill in the right-hand column with the description of what each organ does. To help you, we'll show a clip that explains the process of digestion. But first, pause the tape and copy down the table. Here comes the video clip.
Saliva in the mouth begins the job of breaking down the carbohydrates like starch. The part-digested ball of food is swallowed and squeezed down the esophagus towards the stomach in a wave-like motion called peristalsis. It takes about 6 seconds for fairly solid food to go down, less than that for liquids. In the stomach, the food is mixed with digestive juices containing enzymes and hydrochloric acid, which is secreted from the glands in the stomach wall. These begin to break down the proteins. The part-digested food is squeezed into the small intestine through the duodenum. Digestive juices from the pancreas continue the breakdown of starches. Bile from the liver emulsifies the fats. Inside the small intestine, villi increase the surface area and aid absorption of small molecules into the bloodstream. By the time the mixture leaves the small intestine, almost all of the digestive food will have been absorbed through its walls. The rest travels very slowly through the colon, where the last stages of absorption take place. What is left is largely fibre, mixed with bacteria in the colon. This is finally expelled through the anus.
If you need some more help, here's a diagram of the human digestive system. You can also find it in the biology section of the GCSE Bitesize website. By now, your completed table should look something like this. Food enters the mouth, where chewing reduces it into small pieces, increases its surface area and mixes it with saliva. In the stomach, food is mixed with gastric juices containing hydrochloric acid and the enzyme pepsin, which begins to break down the proteins. The part-digested food is then squeezed into the duodenum, the first part of the small intestine. Here, enzymes from the pancreas digest starches, fats and proteins. Bile made in the liver comes from the gallbladder and emulsifies fats. The pancreas makes enzymes that are added to food in the duodenum. The ileum is the rest of the small intestine, where digested food molecules are absorbed into the blood by diffusion through the villi. The colon is the large intestine that slowly moves food onwards. Most of the water and salts are absorbed by osmosis from undigested food. Undigested food is passed down to the anus. It's egested. Let's look at the function of enzymes in more detail. Enzymes are important in the digestive process, but how do they work? Most digestion processes are chemical reactions that involve large molecules of carbohydrates, proteins and fats. Enzymes speed up their breakdown into smaller, soluble molecules, which can be absorbed more easily by the body. All enzymes are catalysts and speed up chemical reactions. They're specific, which means they work only on one substance or type of food. They're temperature sensitive and work best within a narrow range of temperatures. Enzymes involved in human digestion work best at body temperature around 37 degrees Celsius. They are made of proteins and begin to be destroyed at temperatures much above body temperature. They work best within a narrow range of pH, that is, in either acid, alkaline or neutral conditions. The three types of enzymes involved in human digestion are amylase, protease and lipase. Amylase changes carbohydrates into glucose. Protease changes protein into amino acids. And lipase changes fats into fatty acids and glycerol. This is the end of the section on human nutrition. This section is about human blood circulation. It covers how the heart circulates blood around the body and the composition of blood itself. First, we'll look at how the heart circulates blood.
The heart is a complex muscle. It beats 3,000 million times in a human lifespan, forcing the blood round the body once every minute.
If we look inside the heart, then we can see there is a thin-walled chamber and a thick-walled chamber. The smaller chamber delivers blood through the pulmonary artery to the lungs for oxygenation. It then returns to the thick-walled chamber to be delivered to the body through the aorta.
Fresh blood leaves the heart through the aorta, the first part of a system of arteries that branch out all over the body. Because they're under pressure, arteries have thick walls. From the arteries, the blood squeezes through capillaries. Their walls are so thin that nutrients and oxygen can diffuse out to feed cells, while waste products move into the bloodstream to be carried away. The dirty blood is collected in veins. As it's now at low pressure, veins have quite thin walls and valves to make sure the blood flows the right way back to the heart. But before it goes round the body again, the heart pumps the blood in a short second loop through the lungs. Because of that second loop, the human circulation system is called a double circulation.
So, to summarise, arteries are thick-walled muscular tubes that carry blood away from the heart. Veins are thin-walled tubes that carry blood back to the heart. They have a large diameter and valves to keep the blood flowing in one direction. Capillaries are very narrow tubes with walls only one cell thick, which carry blood through our tissues. But how does the heart actually work? The vena cava is the main vein that brings blood from the body to the right atrium. The blood goes through the tricuspid valve into the right ventricle. It's then pumped out through the pulmonary artery to the lungs. In the lungs, the blood is enriched with oxygen. Blood that's rich in oxygen then comes back through the pulmonary vein into the left atrium. It then goes through the bicuspid valve into the more muscular left ventricle, which pumps it off to the whole body through the aorta. See how the heart is doing two things at once? That's called double circulation. While the right ventricle is pumping blood to the lungs, the left ventricle is pumping blood right around the body. The left ventricle wall is more muscular than the right ventricle because it has to push the blood further around the body. You need to be able to label the various parts of the heart properly. Remember, the left and right on the diagram are the opposite way to your own left and right. They're as if you're looking at the heart from the front. This diagram is also in the biology section of the GCSE Bitesize website. Now, what about the composition of the blood itself? Blood is a liquid medium that transports dissolved substances around the body. Blood is made up of plasma, red and white blood cells, and platelets. Plasma is a straw-coloured watery fluid. It carries digested food particles like glucose and amino acids and dissolved mineral salts, hormones and dissolved gases such as oxygen and carbon dioxide, waste products such as urea, the white and red blood cells and platelets, and antibodies produced by the white blood cells. Platelets are fragments of cell which help the blood to clot after a cut or injury. Red blood cells carry oxygen to all the body's cells for respiration. They have a red pigment called hemoglobin, which combines easily with oxygen. This makes oxyhemoglobin, which gives up oxygen easily wherever it's needed. Red blood cells are small and flexible, so they can pass along capillaries to carry oxygen to the cells. Unlike most other cells, they have no nucleus. White blood cells engulf and digest foreign bodies like bacteria. They are adapted to their function because they change their shape to flow around bacteria. White cells also make antibodies to aid the body's immune system. Antibodies fight threats from viruses and infections from outside the body. The next clip explains how antibodies and the body's immune system works.
Normally, the body's immune system has a whole army of antibodies that protect it against invading cells. Any unfamiliar cells are dealt with by the antibodies. T helper cells assist by sniffing out unfamiliar bodies in whatever form they take. They warn the immune system to make the right kind of antibodies. New antibodies destroy the invader. These antibodies remain on standby in the body in case the invaders return.
That's the end of the section on blood circulation and the heart. This section is about breathing. When you breathe, you inhale oxygen and exhale waste gases. Breathing is the process of ventilating the lungs with air. Inside the lungs, blood absorbs oxygen from the inhaled air and circulates it around the body. The lungs also absorb carbon dioxide from the blood in the lungs and exhale it into the air. But how do they do it? Here's a diagram of the lungs. This is how ventilation takes place. During inhalation or breathing in, the intercostal muscles move the rib cage upwards and outwards. The diaphragm contracts and becomes flatter and less dome-shaped. That increases the volume inside the thorax, that's the chest cavity, making the pressure inside lower than the atmospheric pressure outside. That makes air rush into the lungs. During exhalation or breathing out, the rib cage moves downwards and inwards. The diaphragm relaxes and becomes more dome-shaped. That decreases the volume inside the thorax, making the pressure inside higher than the pressure outside. That makes air rush out of the lungs. Air travels to and from the lungs along the trachea. The trachea divides in two branches, called the bronchi, which lead to the left and right lungs. Each bronchus divides again and again to give a mass of tiny tubes, called bronchioles. They all end in tiny air sacs, or alveoli. Let's look at that in real life, and follow the air into the lungs to see where it goes.
It's possible to look inside the lungs and airways to check for damage by putting a camera down through the nose.
But you shouldn't feel any pain at all. I'm just going inside the nose now, and it's a bit difficult to get a decent view to start with. As we just go inside there, we're going towards the back of the nose, and in a second will appear at the back of the nose. Here we are, going out of the back of the nose now. And as I explained, the back of the nose joins up with the back of the mouth. So I know exactly where we are on the screen now. We're looking at the back of your throat from the inside. And as we move down a bit, we'll begin to see the entrance to your voice box. This thing which is going horizontally across the screen is the epiglottis. It's like a little valve which flaps down over the voice box when we're swallowing, so that food goes down the gullet, the esophagus, into the stomach and doesn't go down into the windpipe. That's lovely. And through the vocal cords we go into the windpipe. Now the windpipe, as I can see it here, is reinforced with rings of cartilage. You can actually see those, in fact they're horseshoe-shaped rather than ring-shaped. Horseshoe-shaped cartilage. That's fine. And we've got an excellent view now. Just here we've got the division into your right lung and your left lung. And we can see how the tubes actually branch. Just now you can see it dividing and subdividing as we go look into all those little holes. That all looks absolutely fine. And I then go on down towards the bottom of your right lung. So I'm actually looking down here now. Right lung, I'm looking right down the bottom of your right lung now. And I can see these various tubes dividing and subdividing. It all looks beautifully normal. A nice non-smoker's lung.
Make sure you know how to label all these parts, in particular the trachea or windpipe, the bronchus, the bronchioles, the alveoli or air sacs, and the actual mechanisms of breathing. This diagram is also on the GCSE Bitesize Biology website. But what exactly happens inside the alveoli in the lungs? As the air enters the alveolar air sacs, oxygen diffuses through the thin walls into the circulating blood and is carried off around the body. But that's not the only gas exchange that's happening. Blood returning from the body is high in carbon dioxide. That diffuses the other way, from the blood and into the air sacs, and is then excreted from the lungs when you breathe out. It's important to remember that both these gas exchanges happen at the same time. So the main function of the lungs is the exchange of gases, absorbing oxygen and excreting carbon dioxide. Both these processes take place in the alveoli, or air sacs, which are richly supplied with blood. That's the end of the section on human breathing. The next section is about respiration. It's important not to confuse respiration with breathing. Breathing is the ventilation of the lungs which allows gas exchanges to take place. It provides oxygen for respiration and removes the carbon dioxide that's produced. Respiration is the process of transferring or releasing energy from food. Respiration happens in a series of chemical reactions that take place inside each cell in the body. The word equation for respiration is glucose from food plus oxygen from inhaled air gives carbon dioxide plus water plus energy. Glucose sugar is oxidized in all the cells of the body, and the cells use the energy released. The byproducts are carbon dioxide and water. Respiration in the presence of oxygen is called aerobic respiration. There's more about human respiration in the higher tier science programme. That's the end of the section on respiration. This section is about the human nervous system. This section covers the reflex arc, the eye, and defects of vision. First, the reflex arc. The nervous system works like an internal communication system. It uses our senses to detect stimuli from our environment, such as light, heat, sound, smell, taste, or gravity. Most of these stimuli are then transmitted to the brain. In the brain, the stimuli are translated into messages to other parts of our body. But not all the nervous system works that way. Reflexes are responses in our bodies which happen automatically without a stimulus being sent by the brain. This process is called a reflex arc. Examples of reflexes are pulling your hand away from something painful, the way your knee jerks when it's hit lightly on the tendon below the knee, and changing the size of the pupil in your eye in bright light. This clip looks at some reflexes.
When we're alive, our eyes respond to light, even when we're unconscious. The pupil doesn't change size if the brain is dead. The blink reflex is another indication of life, but some of our other senses are much more difficult to test.
Although we can't test hearing in an unconscious patient, the same nerve serves the sense of balance. And this can be tested by bathing the ear with ice-cold water to see whether the eyes move. We can sense touch by pressing on the fingers and the toes. The sense of taste also can't be tested in someone who is unable to speak, but we can touch the tongue and around the mouth to see if the patient swallows or tries to gag. We also examine whether or not the patient is able to take independent breath.
But how does the reflex arc work? If you touch a hot plate, receptors in your fingers sense the heat. The stimulus goes along a sensory neuron as an electrical impulse to the central nervous system in the spinal cord. There, the signal passes from the sensory neuron to a relay neuron across a junction called a synapse, using chemicals called neurotransmitters. The relay neuron passes the impulse on to a motor neuron, and the motor neuron carries the impulse to an effector, such as a muscle which takes action and moves your hand. Remember, all this happens very quickly. It's quite automatic and no decision making is involved because the reflex arc does not involve the brain. To summarise the reflex arc, sense organs detect stimulus. Impulse carried along sensory neuron, which passes from sensory neuron via relay neuron to motor neuron. Motor neuron carries the impulse to effector muscle. Effector muscle then takes action. Remember, all this happens very quickly. It's automatic and no decision making is involved. The next section is about the human eye. The eye is one of our most important sense organs. It's sensitive to light and allows us to see. How does it work? As you watch the next clip, see how each part of the eye responds to light.
At the front of the eye is the cornea. It's clear like a window. Behind the cornea sits the iris, a round disc that blocks light, except for a hole in the middle called the pupil. The pupil controls how much light comes into the eye. Next is the lens. It's clear like the cornea, but its shape can change. Because the lens can change shape, it can focus light from objects both near and far away. The light ends up projected onto the retina, a light-sensitive layer of cells at the back of the eye. When the lens is relaxed, it's thin and flat. This brings the light from distant objects into focus on the retina. But when the muscles around the lens contract, the lens becomes fatter and brings the light from close objects into focus. The lens accommodates the changes of focus from close to distant objects by relaxing and contracting to alter its shape. This is why it's called accommodation. The retina that covers the back of our eyes is made from a layer of cells that are sensitive to light. So the retina is a bit like the film in a camera. The lens projects an upside-down image onto the retina, and the light-sensitive cells convert each patch of it into an electrical signal. Nerves carry that signal to our brains, but their wiring is arranged so that we see the world right way up.
Let's summarise. The iris is the coloured part of the eye. The pupil is the hole in the iris which lets light into the eye. The muscles in the iris control the amount of light entering the eye by changing the size of the pupil. Light then passes into the eye through the clear lens. The shape of the lens is controlled by the ciliary muscles. The lens focuses light onto the retina. The retina is a light-sensitive layer lining the back of the eye. The retina changes the light image focused on it into nerve impulses. The nerve impulses go to the brain to be translated into what we see. Now we'll look at some defects of vision.
Imagine this glass bowl is the eyeball. There's a lens at the front, and water with green dye shows the path of the light rays. When a normal eye looks at a point of light, like a candle, the lens bends light rays so they converge, and we see clear, sharp images. But for some people, the lens doesn't match the size of the eye. Sometimes the light rays focus short of the retina, like this, making a blur. This is called short-sightedness. In other people, the light rays don't converge at all. This is called long-sightedness. To focus properly like this, short or long-sighted eyes need some outside help, spectacles or contact lenses. The extra lens moves the point where the light rays converge, back to fix short sight and forward to fix long sight.
So people with short sight can see close objects clearly because the lens can focus a clear image on the retina. They can't see distant objects clearly because their lens can't focus the light onto the retina. The clear image falls short. Short sight is corrected by using a concave lens in glasses or contact lenses. That moves a clear image back so it focuses on the retina. People with long sight can see distant objects clearly because their lens can focus a clear image on the retina. They can't see close objects clearly because their lens can't focus the light onto the retina. The clear image falls behind it. Long sight is corrected by using a convex lens in glasses or contact lenses to move the clear image forward so it focuses on the retina. That's the end of this section on the human nervous system. This section is about human hormones. Communication between various parts of the body is maintained both by the nervous system and by hormones. Hormones are chemical messengers that pass round the body and bring about changes in the body. Hormones are produced by glands in various parts of the body. The pituitary gland controls growth rate, menstrual cycle and lactation. The thyroid gland produces thyroxine, which controls the metabolic rate. The adrenal gland produces adrenaline and affects our fight or flight responses by increasing heart rate and blood sugar levels and directing blood to muscles and brain. The pancreas produces insulin, which controls blood sugar levels. The testes produce testosterone, which causes male puberty changes and stimulates the production of sperm. The ovaries produce estrogen, which causes female puberty changes and helps control the menstrual cycle. Hormones are produced by glands and secreted into the bloodstream. They are transported around the body in the blood. Hormones affect specific cells, target cells, elsewhere in the body. Generally, hormones have slow, long-lasting effects. Adrenaline and insulin are two hormones that have more rapid effects. Now we'll look at two hormones in detail, insulin and sex hormones. First, insulin. The next clip is about insulin. As you watch it, note down the ways it controls the sugar level in the blood and what happens when it doesn't.
Insulin is produced by an organ called the pancreas, which is found just behind the stomach. The white specks are made up of cells which produce insulin. The function of insulin is to enable sugar in the form of glucose to pass from the blood into the cells. If insulin is absent, glucose can't enter and the cells are starved of fuel. Sugar accumulates in the blood. Because the muscles are starved of sugar, the body makes energy from its fat and protein reserves, so the person loses a lot of weight.
You should have noticed that insulin is produced naturally in the pancreas to control the level of sugar in the blood. It helps sugar in the form of glucose pass from the blood into the cells. It also controls the level of sugar in the blood by increasing the rate at which the liver can convert sugar into glycogen. When the blood sugar returns to normal, insulin production stops. Diabetes is an illness which occurs when the body doesn't produce enough insulin to keep its glucose level steady. Low insulin production starts a series of reactions. Symptoms of diabetes include a high level of sugar in the blood. Glucose is no longer turned to glycogen or fat to give the body energy. Diabetics lose weight and excess glucose is excreted in the urine. This needs a lot of water, so diabetics feel very thirsty. Diabetics can also become unconscious unless they are given artificial insulin.
Covering like that.
It's important that diabetics check their blood sugar levels regularly.
And you press the timer there, so you can see that's 23.4, Louise, and that's far too high at the moment. But it's nothing to worry about because the doctor will just give you some more insulin tonight and bring it down. Let's compare it against this colour chart.
Because diabetes has been known about for a very long period of time. In fact, it's been recorded back at the time of the Greeks. And when they referred to diabetes, because it was a wasting disease, you passed so much urine that you had to drink a lot and you lost a lot of weight. And in those days, you would die from the disease because you weren't able to correct it. And it wasn't really until 1921 when probably the most famous couple who won a Nobel Prize, Banting and Bess, first described the origins of diabetes in the pancreas. What they actually did was, and some previous co-workers, removed the pancreas from dogs, in fact. And on removing this pancreas, the dogs immediately became diabetic. In other words, what they started to do is drink a lot and pass a lot of urine, and ultimately they would die within a few weeks. They then found that by taking extracts of this pancreas and mixing it up in a laboratory situation and then giving it back to the animals, that they corrected the problem of diabetes and they would be back to their normal selves.
When insulin was injected into human diabetics, the results were dramatic.
Next, sex hormones. Make sure you know the role played by the human sex hormones. These are estrogen and progesterone in women and testosterone in men. The ovaries produce the female sex hormones estrogen and progesterone. They're responsible for sexual development and the menstrual cycle. The testes produce the male sex hormone testosterone. It controls male sexual development and the production of sperm. Here's a sample question about human hormones. Explain how insulin helps to control the level of glucose in the blood. Stop the tape and have a go at this. I'll look back over the earlier sequence. How did you get on? A simple answer is that insulin makes the level of glucose go down, but that's only worth 1 mark because there's only one fact. The pancreas secretes insulin, which makes the liver turn glucose into glycogen, might get 3 marks as it makes 3 correct points: that the pancreas makes insulin, that insulin affects the liver, and that the liver turns glucose into glycogen. A full answer would be something like this. The pancreas secretes a hormone called insulin when there is too much glucose in the blood. The liver is the target organ, which turns the excess glucose into glycogen. When the blood sugar level returns to normal, negative feedback turns the secretion of insulin off. That's the end of the section on hormones. This section is about homeostasis. Homeostasis means keeping our body's functions in a steady state so everything works properly. We're going to look at how the body regulates temperature and how the kidneys control excretion and osmoregulation. That's how water levels in the blood are regulated. First, how do we control our body temperature? This next clip takes a look at one method. Can you think of any others?
Evaporation happens when molecules of water try to turn into vapour. To do that, they must break free from the surface of the water. And breaking free takes extra energy. If the water is resting on warm skin, the nearest available energy is the heat energy from that skin. So the molecules take the energy they need from the skin and break free. That leaves the skin with less heat, so it cools down. This is why we sweat when we get hot. A microscope reveals the body's cooling system. Tiny glands in the skin which can let out water from the blood. Once the water's evaporated and the body's cooled down, the glands stop sweating automatically.
There are other ways the body controls our temperature. If the body gets too hot, blood capillaries dilate to increase the flow of blood to the skin to remove heat. Also, hair muscles relax, so the body's hair lies flat and heat can escape. Too cold, and the capillaries constrict to reduce the amount of heat being lost from the blood through the skin. And the hair muscles contract, making the hair stand up, trapping a layer of insulating air to keep the body's heat in. This animation is also on the GCSE Bitesize Biology website. Draw up two columns with the mechanisms that raise the body temperature in one column and the mechanisms that lower the body temperature in the other. Don't forget, you can always go back and have another look at the animation. So, how did you get on? Mechanisms that raise the body temperature include vasoconstriction, where the blood vessels near the skin surface narrow to reduce the flow and loss of heat from the blood. Shivering, that's rapid involuntary contraction of the muscles to produce heat. And fat stored for insulation. There's also heat gain by contact with something warm. Mechanisms that lower the body temperature include vasodilation, where the blood vessels near the skin surface dilate to increase the flow of blood and loss of heat from the blood. The evaporation of sweat, which causes cooling. Panting in animals, which causes cooling by increasing evaporation from the tongue. And loss of heat by contact with something cool. There's also natural heat radiation from the whole body surface. This next part of homeostasis looks at excretion. Excretion means getting rid of metabolic waste, which the body produces as a result of the different reactions that are continually taking place in it. The two major organs involved in excretion are the lungs, which excrete carbon dioxide and water, and the kidneys, which excrete urine. The skin also excretes water, salts, and some traces of urea as sweat. First, let's see how the lungs excrete carbon dioxide. In the section about human respiration and breathing, we saw how all cells respire and produce carbon dioxide and water. Carbon dioxide is a waste product and is transported in the blood to the lungs, where it is diffused into the alveolar air sacs and excreted or breathed out. Next, how do the kidneys excrete urine? When proteins are metabolized in the body, urea is produced. Urea is poisonous to the body tissue and must not be allowed to accumulate. It gets dissolved in the blood and is excreted by the kidneys along with the excess water and salts as urine. Next, osmoregulation. How the water levels in the blood are regulated.
Only a third of the human body is made from solid material. 2/3 is water. Blood and body fluids make up about 45 litres in an adult. Every day we pass out a couple of litres in urine. Sweating takes up to another litre, and moisture in our breath uses still more. The exact figures depend on what the body is doing, but on average, doctors recommend we drink about 3 or 4 litres a day. Drinking too much normally does no harm. The water the body doesn't need is turned into urine by our kidneys.
We have 2 kidneys, and they're situated in our back, just at the bottom of the rib cage on the back, on either side. This comes from a post-mortem. It's the human kidney, and here is the artery, which takes blood from the body, and the kidneys filter the blood. Once the blood is filtered, it passes back through a vein, just underneath the artery, and the filtering process produces urine. The urine gets concentrated in this part here, and then it is passed down the ureter, this tube here, to the bladder. And eventually we feel the need to pass urine.
So the kidney is responsible for regulating the water concentration in the blood, as well as excreting urine. There's more about homeostasis in the higher tier science programme. That's the end of the section on homeostasis. This section is about human health. In this section, we'll look at the effects that cigarettes and alcohol have on the human body. First, cigarettes. Smoking cigarettes affects the brain. It causes heart disease and damages the lungs and airways. As you watch this clip, note down the different ways that cigarettes damage human health. Cigarettes contain nicotine, which is highly addictive and makes you want to smoke more. But that's not all.
There are many other chemicals in cigarette smoke, and they are grouped together as tar. Each one of these flasks contains the dissolved tar from just one cigarette.
This is an example of tar. This thick, black, treacly substance has over 4,000 chemicals in it. We know that it causes cancer. We know that it causes a narrowing of the airways into the lungs. We know that it causes an increase in mucus production. And we certainly know that it damages the little hairs that line the lungs, which help to protect the lungs from dirt and diseases.
The effect of smoke on the lungs is very dramatic.
Here are the contents of the chest, the neck organs, the heart in the middle in its sac, and on each side the lungs. Now, even the normal lung, we can see, is black, with all the soot that he's inhaled with that smoke over the ears. And the affected lung is even blacker still, and the bottom of the lung is solid. Whereas the normal lung is quite squidgy, this affected part of the lung is quite solid. It's stuffed with pus.
Smoking also causes lung cancer.
You can see that there's the blood-filled tubes on either side. We've got this white mass of tumour, so that when my doctor friends looked down a little telescope, they could see that this was growing through the wall of these air tubes here. And that's what was giving him his cough, and it was that which was causing this bleeding.
We saw that most damage is done to the lungs by cigarette tars. They can narrow the airways into the lungs, making breathing more difficult and cause cancer of the lungs themselves. Tars increase mucus production. This causes coughing, which damages the tiny hairs, the cilia, that keep dirt and dust out of the lungs. Smoking cigarettes also causes emphysema, in which the lungs' air sacs are damaged by tar and stop working properly. Smoking also causes heart disease because it damages the arteries.
Another major cause of death for smokers is heart disease, where the arteries become blocked.
Smoking cigarettes accelerates aging of the arteries and disease in them, and so even quite young people can end up with coronary artery disease and die of it. We are going to take a vein from his leg and an artery from inside the chest wall to bypass the blockages in his arteries, and this will remove his chest pain and hopefully make him live longer.
What effect does alcohol have on the human body? During the next clip, see how alcohol can damage human health.
In this unit here, we perform tests on all sorts of new drugs. They're administered to human volunteers for the first time after they've been tested in animals. There is absolutely no doubt that alcohol would not pass these tests in volunteers. It has harmful effects both on the liver and on the tummy, for example, which would mean it would not be passed to be given out to the general public.
People who drink notice the first effects of alcohol on their brain. Their behavior changes because alcohol affects this part of the brain, causing them to feel often cheerful. It's a surprise to many people to know that alcohol is a sedative because of the way they feel. But this sedation actually stops you behaving in a way that you normally would.
It's not only our brain that is affected by alcohol. It has an effect on our whole body. When we take a drink, swallowing causes a flap in our throat called the epiglottis to close, allowing the liquid to go into our stomach instead of our lungs.
If one is sick, particularly with alcohol in the vomit, some of it gets into the lungs and can cause a lot of damage. So this mechanism is important. It's important to remember, too, it can be suppressed by alcohol. After swallowing, the fluid runs down over the back of our tongue and gets into the esophagus, the gullet, down it goes, through the diaphragm about here, and then into the stomach. And the stomach is where the process of absorption and digestion begins. Now this stomach is empty. Whether your stomach is full or empty makes a big difference to how quickly you absorb drinks. And in particular, if the drinks are fizzy, because they tend then to be acid, they tend to pass through the stomach very much more quickly and be absorbed very readily. After the stomach, we go through the duodenum, and absorption then takes place in this tightly coiled intestine, the guts.
Drink is absorbed by tiny finger-shaped villi inside the gut.
You begin to absorb alcohol within a few minutes of drinking it, and most of it is absorbed within about 45 minutes or so, assuming you've drunk on an empty stomach. The alcohol, which has been absorbed through the gut, goes via a large vein to the liver, entering the gland about here. It then circulates in the liver, and the liver cells break down the alcohol, the blood returning to the circulation through these large veins. The liver can only break down a certain amount of alcohol on each time that the blood passes through. And this rate is quite slow. You can still be drunk the morning after a party. This is a slice of a normal liver. It has an even brown surface, a regular pattern of blood vessels, and a uniform structure. This slice of liver is from a patient with cirrhosis. Cirrhosis is a condition that can affect those who drink alcohol to excess, and you can see the difference clearly on the surface of the gland. Instead of an even pattern, you have lots of little, we call them nodules. They're areas where the liver cells are growing in an attempt to make up for the damage done to them on previous occasions when alcohol's been taken. The blood leaves the liver, still containing alcohol, and comes to the heart. Now this is a preserved specimen of heart, which enables me to show you what a strong muscular pump it is. This pump pumps the blood on into the lungs and to the rest of the circulation. Those who drink alcohol for a long period of time, in some instances, can damage the working of this pump as well.
So alcohol can affect the brain, distorting people's behaviour by removing their inhibitions. It can impair coordination, which is why drinking and driving is so dangerous, and it causes damage to the stomach, heart and the liver. That's the end of the section on humans as organisms. This section looks at green plants as organisms. It's divided into nutrition, the role of hormones, and the transport of water in plants. First, nutrition. Plants convert carbon dioxide and water into food in the form of carbohydrates. That process is called photosynthesis. Photosynthesis is an essential process, not just for plants, but for all life on Earth. As you watch the following clip, note down the substances that plants use up during photosynthesis and what they produce while they're doing it.
Photosynthesis is something no animal can do. Carbon dioxide from the air seeps into the leaves through pores, called stomata. It reaches tiny granules, chloroplasts, that contain a green chemical, chlorophyll. Chlorophyll is the key to photosynthesis. It uses energy from sunlight to bond the carbon dioxide to the hydrogen in water brought up from the roots. The result is carbohydrates, sugars and starches that the plant can use to grow. But photosynthesis also produces oxygen. Every day, each person on the planet needs to absorb several hundred balloon volumes of pure oxygen from the air. And we each produce several hundred balloon volumes of carbon dioxide. So the Earth's atmosphere is in a constant balance. Plants recycle our carbon dioxide, and we recycle their waste oxygen. Plants, humans, and animals across the planet need each other.
So, during photosynthesis, chlorophyll in green plants bonds carbon dioxide, taken in from the air, and hydrogen from water. Plants use the energy from sunlight to convert the carbon dioxide and water into carbohydrate and oxygen. The carbohydrate is in the form of glucose, some of which is used as food, but the rest is converted to starch and stored. The oxygen is given off and used for respiration. This is the word equation for photosynthesis. Carbon dioxide and water produce oxygen and glucose in the presence of sunlight and chlorophyll. Sunlight provides the energy, while chlorophyll is the catalyst for the reaction. Now let's think about the rate of photosynthesis. The word rate describes how fast a process is happening in a certain time. The more photosynthesis occurs, the more food is produced and the more growth happens. The amount of living tissue produced is called biomass, normally measured in grams or kilograms. As you watch the next clip, see how growers control the rate of photosynthesis to increase yield or biomass.
5 a.m. At one of Britain's biggest tomato nurseries, it's time for breakfast. Over the next hour, these plants will suck up most of the 50,000 litres of water they will need for photosynthesis today. It's pumped to their roots. This is photosynthesis running at full speed. In nature, the rate of photosynthesis is always limited by a shortage of light, water, or carbon dioxide, or because the plants are at the wrong temperature. But here, everything's controlled to boost plant growth. Since they started, they've managed to push the plants to yield 8 times the crop they would grow in a field. Pat Smith is the production supervisor. She leads a team of 8 full-time tomato pickers.
Well, basically, we give them everything that they need to grow. All the light that they want from the high greenhouses, heat from pipes on the ground, and carbon dioxide from the tubes, which shoots out at little pinpricks at the side, and all they've got to do is grow.
The plants have everything, except soil. This is a hydroponic nursery, and the roots grow directly in running water. Here is living proof that plants can grow out of thin air. Last year, these plants used photosynthesis to turn carbon dioxide and water into 800 tons of tomatoes.
The rate of photosynthesis is normally limited by one or more of the following factors: the amount of light, the temperature, and the amount of available carbon dioxide. As well as photosynthesizing, green plants also respire. Respiration is the process by which energy from food is released within cells. Both food and oxygen are required for respiration, and carbon dioxide and water are produced. At the same time, energy is released. While green plants can only photosynthesize during daylight hours because they need sunlight, they can respire 24 hours a day. The next clip proves it.
Just like animals, when plants respire, they use oxygen in the air and produce carbon dioxide and water. But in the light, plants photosynthesize too. So to investigate the gas exchange and respiration alone, I'll have to do the next experiment in the dark. No light means no photosynthesis. This cylinder passes pure oxygen down this tubing into a bell jar. There's no carbon dioxide in there at all. Another tube passes the outlet gas through this bicarbonate indicator. Left overnight, if the plant is producing carbon dioxide, the indicator will turn yellow. So, sure enough, just like us, this plant is producing carbon dioxide.
Let's compare the equations for respiration and photosynthesis. Photosynthesis is carbon dioxide plus water gives glucose and oxygen in the presence of chlorophyll and sunlight. Respiration starts with glucose plus oxygen and gives carbon dioxide and water with energy released. If you look carefully, you'll see that respiration is the opposite of photosynthesis. Photosynthesis needs energy from the sun to take place. It uses carbon dioxide and water as raw materials. It's a building up process, with larger carbohydrate molecules being made from smaller molecules of water and carbon dioxide. Oxygen is a waste product. Respiration is the opposite. It releases energy and transfers it to the cells. The raw materials it starts with are carbohydrate and oxygen. Respiration is a breaking down process because larger carbohydrate molecules are broken down into smaller carbon dioxide and water molecules. Carbon dioxide and water are the waste products. There's more about plant nutrition in the higher tier programme. That's the end of the section on plant nutrition. This section is about plant hormones. Hormones are chemical messengers that bring about changes in a plant or animal. It's not just humans who have these chemical triggers in their bodies, plants have them too. In plants, hormones influence growth and movement. Tropism is the growth movement in plants. It is triggered by a stimulus in the plant's environment. For example, roots grow downwards in response to gravity. This is called geotropism. Shoots grow upwards in response to light. This is called phototropism. In the next clip, see how the tropism response is triggered, where it happens, and how it works.
Plants grow and reproduce themselves in every kind of environment on Earth. To be able to survive, they have to be able to receive information about the world around them. And they must be able to respond to these signals. If you put a plant on a windowsill, you'd expect it to bend towards the light. And if you plant a fruit or a seed, you expect the root to grow down and the shoot to grow up. As the light source is moved from side to side of these cress seedlings, the shoots grow from one direction to another towards the stimulus, and the plant starts to bend within 10 minutes. Over 100 years ago, in this house in Kent, Charles Darwin was fascinated by his study of plant growth in varying light conditions and performed some simple, now classic experiments. He grew oat seedlings and observed what happened to the coleoptars. He covered the tips of some of the seedlings with tiny metal caps. He found the cap seedlings didn't bend when illuminated from one side. And he noticed too that the untouched plants bent not at the tip, but farther down the shoot. Darwin likened the tip to a primitive brain, sensing the light and then sending some influence down the shoot. Some plant growth substance in the shoot tip is activated by light and passes down the stem to cause elongation in the cells behind the stem tip. This growth promoter, or auxin as it was called, only moved down the shaded part of the stem, and so this grew more quickly than the side receiving the light stimulus, and bending towards the light occurred.
The growth promoters revealed by these experiments are plant hormones. Plant hormones trigger and control growth in response to different stimuli in the environment. Hormones are produced in the tip of the plant's shoots and work by causing the elongation of the cells in a particular part of the shoot. Plant hormones move from the tip back to the rest of the shoot by diffusion. Gardeners make use of the way hormones are produced in plants when they prune them by pinching out the tips of the shoots. This pruning stops the production of hormones and diverts growth into side shoots, so the plant becomes bushier. That's the end of this section on plant hormones. The next process we look at is the transport of water in green plants. The uptake, transport and use of water are vital for the survival of plants. During the next sequence, note down the key points about these three aspects of plants and water.
This jet of water, 70 feet up in the air here, it takes that huge, big, noisy engine down there. But this tree pumps up about 100 gallons every hour and manages to do so in total silence. How? The answer is to be found in the tree's trunk. The central part of this is wood. Around the outside of this pillar, there are ranks of hair-thin pipes. Those immediately beneath the bark carry the food-laden sap down from the leaves. Farther inside the trunk, there's another set of tubes. These are the ones that carry the water up. They are continuous pipes that extend the whole length of the trunk. As the water evaporates in the leaves above, the long, thin threads of it are pulled up the tubes, into the branches, and ultimately into the leaves themselves. Some of it is used in the food-making process. The rest evaporates through the leaf pores as vapor.
So, to summarise, water passes into the plant by diffusion through the roots. It passes up and around the plant in xylem vessels, which are tubes made from dead cells. Water leaves the plant through pores on the surface of the leaves called stomata. This process of water loss through the leaves is called transpiration. Here's a sample question about the transport of water in plants. Explain how and why plants close their stomata when it's hot or windy. How did you get on? One short answer is that plants close their stomata to stop losing water. But that's only worth 1 mark because it doesn't say how plants close their stomata. Also, plants close their stomata with their guard cells only explains how, but not why. A full answer would be something like, plants close their leaf stomata to stop them losing too much water. They do this by letting their guard cells go flaccid. Water can only get out of the leaf through the stomata because the epidermis is waterproof. That's the end of the section on green plants as organisms. This section is about variation, inheritance, and evolution in animals and plants. We'll start by looking at variation. The term variation refers to the slight individual differences within populations. All living things change and evolve from one generation to the next, and as they do so, more variation is introduced. But how does it happen? Plants, just like people, exhibit differences between individuals of the same species. Some of these variations are caused by factors in the environment, but most variation is controlled by the genetic material we inherit. Charles Darwin was a 19th century naturalist who studied variation among plant and animal species during a 5-year voyage around the world.
Darwin's contribution was to provide the big idea, the overall scheme which might explain the complexity of life and how it came to be. Change was at the heart of his theory. Gradual changes from one generation to the next, as the fittest and the best adapted survived and prospered, passing on these favored characteristics. But how were these changes passed down from one generation to the next?
We now know that variation within a species is caused by the environment and by genetics. Environmental factors determine how an organism grows or develops. Genetic variations occur in sexual reproduction because genes are inherited from different parents. Mutation is another cause of variation. Organisms can reproduce either by sexual reproduction or by asexual reproduction. Asexual reproduction does not introduce variation. Only one parent is involved, and asexual reproduction relies on mitosis cell division, which produces identical copies of the original cell. That means offspring have exactly the same genetic information and are exact copies or clones of their parents. Examples of asexual reproduction in plants are sending out runners, as in strawberries, or side branches like busy lizards. Vegetative propagation, such as forming tubers, bulbs, or rhizomes, like in potatoes, crocuses, or irises. Taking plant cuttings or grafts to grow into new plants. And growing a few cells into a complete specimen, called tissue culture or cloning. In sexual reproduction, there are 2 opportunities for variation to be introduced. 2 parents are involved, supplying male and female sex cells, sperm and eggs in humans, and pollen and ovules in plants. They give rise to a new individual by fusing randomly, which introduces variation. Variation is also introduced because sexual reproduction relies on meiosis cell division, which means that each offspring receives slightly different genetic information. That's why we resemble our parents, but are not the same as them. Well, that's the end of this section on variation. Variation leads on to inheritance. Inheritance is the passing on of genetic characteristics from one generation to the next. This was first investigated in the 19th century by Gregor Mendel. As you watch the next clips, make a note of how characteristics are passed on.
Mendel was a monk, later abbot, of the Augustinian monastery in Brunn, a thriving provincial town in what was then a part of the Austrian Empire. Between 1856 and 1863, he raised nearly 30,000 plants, crossing varieties that demonstrated clearly identifiable characteristics, all the while counting the number of times each characteristic appeared in succeeding generations.
What Mendel observed is, for example, if you cross a pink and a white flowered pea plant, then all the offspring in the next generation are pink. So what had happened to the white? And that was the mystery that he started with. Now what happens if we cross two pinks in this generation? Well, that's when the famous ratios pop out in the next generation. You get 3 pinks and 1 white. Now, why is that the case? And Mendel was able to be clear that somehow the white had been masked in the middle generation, but it was still there, ready to pop out at the end. And this is what we would now call the pink being dominant, the white being recessive.
The essence of Mendel's idea is that inheritance is based not on fluids, but on particles. And of course he worked on peas. He worked, for example, on round or wrinkled peas. His amazing breakthrough, really, was to find, and it seems simple, that when you cross a round with a wrinkled pea, you don't get a pea that's partly wrinkled, you get a pea that's round. That pea, if you make those peas together, then you, in the next generation, get the wrinkled peas back again, quite unchanged. So that inheritance is a matter of separate units that are passed down the generations, quite distinct from that which they produce.
Mendel had uncovered a crucial aspect of inheritance. The fact that somehow an individual's characteristics, the color of hair or eye, for example, are passed on through the generations, sometimes showing themselves in a particular individual, sometimes not to be seen again for several generations to come.
It's the same with breeding dogs.
Every characteristic in a dog is controlled by a pair of genes, one from the mother and one from the father. Often the gene from both parents is the same. 2 genes for short hair will make a short-haired puppy, and 2 genes for long hair will make a long-haired puppy. There's no conflict. But when the genes are different, a system comes into play to work out whether the long or the short hair gene will set the hair length. Some genes are stronger than others. They're called dominant genes, and the weak ones are called recessive. Dominant genes tend to hide the effect of recessive genes, sometimes hiding them completely. So if the gene for long hair is completely dominant, a puppy will have long hair, whether it inherits the long hair gene from either one of its parents or from both. The only way it can have short hair is to inherit a short hair gene from its father and a short hair gene from its mother. This means that the effect of recessive genes is often covered up. But the recessive genes are still there, and they're passed on from one generation to the next. With genes for every characteristic of an animal, thousands of pairs of genes are matched up when a baby animal forms. The end result is a complex mix of characteristics from the mother and characteristics from the father.
We saw that genes are responsible for characteristics inherited from parents. Genes can be dominant, which means that characteristic is more likely to be apparent, or recessive, which means that characteristic is less likely to be apparent because it is masked by a dominant gene, although it may appear in later generations. There's more about inheritance in the higher tier science programme. That's it for inheritance. Next, the theory of evolution. In 1859, Charles Darwin published his theory of evolution, based on observation of variation and inheritance in the natural world. As you watch the next clips, note down the evidence that supports Darwin's theory.
Before we go into Darwin's theory itself, we need to be clear exactly what he was trying to do. He was trying to explain not just the origin of species, but also the fantastic variety of species that have ever existed on this Earth. And that brings us to the first kind of evidence, the fossil evidence.
If we look at a sequence of fossils through time, we can see changes and features that have led from one animal group to another. This fossil is a reptile belonging to the group that we think mammals evolved from. This specimen is about 240 million years old. Here are the chewing teeth, and they're relatively simple. It also has a long stabbing tooth here and nipping tooth at the front. If we look at the modern tiger's teeth, we can see here that the chewing teeth have evolved into very strong prong-like structures for chewing meat and bone, as well as having the stabbing teeth and nipping teeth at the front of the mouth. So these gradual changes through time in the teeth and all the other features of the skull actually demonstrate to us that evolution has happened in the fossil record. And what Darwin did was actually find an explanation for how it happened.
Darwin came up with the idea of natural selection.
Very briefly, here's how natural selection works. In every species, there's variations. Some individuals are big, some are small. Some are fast, some are slow. And some of that variation is inherited. So in every generation, most of the children will be born to those animals that have what it takes to survive and reproduce. And those children will tend to inherit what it takes. So as the generations go by, the average animal in the population gets a little bit better. It may seem hard to believe that animals as complicated as these could evolve in the way that I've been saying. But that's because we're not very good at understanding the immensity of time that's been available for it all to happen. Domestic dogs and horses have evolved by artificial selection in a few hundred generations. But life has been going on this planet for about 4,000 million years, and that's plenty of time. When an animal reproduces, its DNA gets passed on to the next generation, that very DNA that made it successful at surviving and reproducing in the first place. Every now and again, rarely, there's a random mistake in the copying of DNA called a mutation. And mutation, together with the shuffling of DNA that happens in sexual reproduction, spreads the variation through the population. Now, if a variation is advantageous, say it gives a predator better eyesight or something, then that's the DNA that will go forward to the next generation. So it's changes in DNA that drive evolution forward.
One of the more surprising things that Darwin suggested in his theory of evolution was that humans and chimpanzees were somehow related.
Now, if Darwin is right, and humans and chimpanzees are very close relatives, then we should expect to see that their DNA is very similar.
Scientists can extract DNA from cells and then cut it up into fragments using special chemical techniques. When these fragments are separated, they can be used to work out the genetic code contained in the DNA. This means that the DNA from different species can be compared.
The order of these fragments of DNA correspond to the order of the different letters of the genetic code. It's much easier to analyze these results on the computer. top line of letters here is what my own genetic code looks like, though it's only a small part of my DNA. We can compare this with the genetic code of a chimpanzee here along the bottom. The two sets of DNA are very similar. In fact, there's only one change here in the stretches of letters that we have here on the screen. If we looked at all my DNA, we would see that 98% of it is identical to that of a chimpanzee. This is very strong evidence that humans and chimpanzees have a common ancestor. The fossil record suggests that this common ancestor was alive 5 million years ago.
The main evidence for the theory of evolution is that the fossil record shows that animals have changed and evolved over millions of years. Over time, most animals and plants have become well adapted to their environment by natural selection. DNA analysis shows that all animal species share a large percentage of identical DNA, so they must have common ancestors. There's more about variation, inheritance and evolution in the higher tier science program. That's the end of this section on variation, inheritance and evolution. This section is about living things in their environment. It's divided into populations and communities, energy and nutrient transfer and cycles in nature. First, populations and communities. The whole Earth is an ecosystem on a global scale called a biosphere. Within the biosphere are many different ecosystems. While watching the next clip, make a note of the different ecosystems you recognise.
Our planet, the Earth, is, as far as we know, unique in the universe. It contains life. Even in its most barren stretches, there are animals. Around the equator, where those two essentials for life, sunshine and moisture, are most abundant, great forests grow. And here plants and animals proliferate in such numbers that we still have not even named all the different species. Here, animals and plants, insects and birds, mammals and man live together in intimate and complex communities, each dependent on one another. 2/3 of the surface of this unique planet are covered by water, and it was here indeed that life began. From the oceans, it has spread even to the summits of the highest mountains, as animals and plants have responded to the changing face of the Earth.
The ecosystems you should have spotted are desert, tropical forest, sea and mountain. In an ecosystem, there's a balance between an environment and the populations and communities of plants and animals living there. The different plant and animal populations are adapted to their environment. This balance is maintained because all the living things are linked through food chains. Sometimes, however, these food chains can be interrupted. In the next clip, we look at a lake in Devon to see why the fish populations, particularly the pike, are decreasing. Work out the food chain in the lake, ending with the pike, and decide why the fish populations are decreasing, as a result of heavy predation, pollution, or other factors.
Slapton Sands, South Devon. A 2-mile-long shingle ridge thrown up over thousands of years by the action of the sea. Behind the ridge is Slapton Lee Nature Reserve, an area of quiet beauty with many rare plants and animals. The main feature is a large freshwater lake, but the reserve also contains a wide variety of different places to live, different habitats, ranging from the open water to reeds, marshes and dense woodland. There are a surprising number of conflicts in the nature reserve. For instance, the fishermen themselves are far from happy. The fishing is definitely not what it was. The changes could be quite natural, or they could be the result of pollution. There's no easy answer. What is clear is that the balance of nature in the underwater world of the Lee is very complicated indeed. If the fishermen are right, the balance in this underwater world has changed significantly in the last few years. The main fish in the Lee are perch, rudd and roach, as well as the pike. The questions are how and why their numbers are changing. And in particular, from the fisherman's point of view, why are big pike now so rare? Some of the answers are slowly being discovered through a detailed study of the balance of nature in the Lee, a study of all its plants and animals, its ecology. Even apparently clear water is teeming with life. These are collections of microscopically small plants. They float around in the water, taking in sunlight and making food. The tiny plants are the source of food for tiny animals, plant-eaters, herbivores. These ferocious-looking insects eat other insects. They are flesh-eaters, carnivores. But they themselves might be eaten by even bigger carnivores, like the perch, and the chain doesn't stop here. The perch are the prey of the much larger pike, the top predator in the lake, the king of this particular jungle. Several theories have been put forward to explain why the pike are smaller than they used to be. For instance, pike feed on perch, rudd and roach, but these fish have become much smaller. It's been suggested that the pike find the small fish a very poor source of food. Each one isn't much of a meal for a hungry pike. Another theory is that there are simply too many pike. They compete with each other for food and space.
You should have worked out that the pike feed on smaller fish, perch, rudd and roach, that themselves feed on insects, who feed on microscopic plants. What happens to the pike if the perch, rudd and roach have less food? If the perch, rudd and roach have less food, they won't grow so big and their population will shrink. As their population shrinks, there will be less food for the pike, which means they will get fewer and smaller as well. When there's a shortage of prey, the predator numbers go down as well. Besides shortage of food, the pike also face competition from other pike for the same amount of food, space, and the opportunity to reproduce. Now for a sample question. Think about a habitat close to where you live. How is that habitat changing and why? Should we be trying to intervene to prevent change? The examiners expect you to be able to apply the principles of conservation to different habitats. So stop the tape and have a go. Some factors you should have noticed about your local habitat are the size of different populations and the relationship between them, like how they feed on each other. You should note the impact of any natural environmental changes and the impact of any human activity. That's the end of the section about populations and communities. This section is about energy and nutrient transfer. A food chain describes the feeding relationship between living things. It shows what eats what. In a food chain, One group of organisms in the chain provides food for the next. A food chain always starts with a green plant, a food producer, and contains at least 3 links. The food chain in the lake at Slapton Leas starts with plants, called producers because they produce food in the first place by photosynthesis. The plant producers are eaten by primary consumers in the chain, tiny herbivores that eat only plants. These herbivores are eaten by the secondary consumers in the chain, flesh-eating carnivorous insects. They in turn are eaten by tertiary consumers, the perch, rudd and roach. Finally, these fish are eaten by a predator, another fish, the pike. Animals all get their energy by eating food, so the flow of energy in a food chain is from the food to the feeder, in the direction of the arrows. All food chains must start with plants, because only plants create their own food by photosynthesis. Without green plants, no animal life can exist. In an ecosystem, food chains do not usually exist in isolation, but are linked together to form a food web of plants and animals that are interdependent. This diagram shows a food web for a seashore community. Here, limpets and sea urchins both eat algae, and the herring gull eats both crabs and sea urchins. Remember, the arrows show the flow of energy around the food chain, which goes from the food to the feeder, from producer to consumer to predator. But that flow of food and energy can't go on forever. The next clip explains why not, and what happens to the energy in a food web.
Deep in the cells of all plants, there's a tiny reaction going on, and basically it's this. Sunlight, filtering through the green pigment, chlorophyll, focuses on a gas, carbon dioxide, and a liquid, water. It shuffles their atoms, making a solid, carbohydrate. And that's fuel. That's stored sunshine. Plants make fuel, but animals can't. So their only choice is to take the plant's fuel by eating them. Animals can eat other animals and get their fuel that way. So the stored sunshine passes from creature to creature. It's a food chain, a fuel chain. And at some point, the chain stops. That's because eating is a pretty sloppy way of transferring fuel. Each animal in the chain only passes on 10% of its energy. The rest is simply burnt off as heat. It doesn't take long to get to the point where the fuel runs out, with an animal that's not worth eating. Here the sunshine stops with the otter. And that's why otters and other big predators are rare.
One way of looking at the amount of energy passed along a food chain is with a pyramid of numbers. That's the number of each animal or plant that the food chain can sustain. For example, it takes 2,000 grass plants to feed 25 voles, which support just one owl. But the pyramid of numbers can be confusing. In this food chain, one large oak tree supports thousands of caterpillars, many blue tits and one sparrowhawk, so it's not actually a pyramid at all. If we change it to a pyramid of biomass, that's the actual mass of biological material at each stage, then one oak tree represents a huge amount of biomass. The caterpillars less, the blue tits less still, and the sparrowhawk even less. So now it's a pyramid. The biomass pyramid also gives a clearer picture of how inefficient a food chain is. In the same way that energy is lost at each stage of the food chain, so the total biomass reduces as you go up the pyramid. Here's a common exam question. Explain what happens to the energy at each stage of a food chain, and why eventually there isn't enough to pass on to a larger animal. Stop the tape and have a go at this question. Remember, the question is asking what happens to the energy, not the food. A fairly full answer would go something like this. To start with, plants make food material by photosynthesis using energy from the sun. When plants or animals are eaten, only some of the food energy is permanently absorbed into the body of the eater. The transfer of energy at each step in a food chain is as little as 10%. The rest of the energy is lost to the surroundings as heat through respiration, excretion and digestion. A food chain rarely has more than 5 or 6 links, because by then there isn't enough energy left to pass on to any larger animal. What if the exam asks you about food webs and habitats you haven't come across before? Well, don't panic. There's always enough information in the question for you to apply what you know about another food web and to answer the question. That's the end of the section on energy and nutrient transfer. This section is about cycles in nature. One important cycle in nature is the carbon cycle. Carbon exists on Earth in many forms: in carbon dioxide gas in the air, as carbon molecules in organic materials such as plants and animals, and in fossil fuels like coal, gas and oil. All these forms of carbon are continually recycling from one to another, but the total amount of carbon remains constant. This is how it works.
The level of carbon dioxide in the air depends on many different processes. Photosynthesis uses carbon dioxide as a raw material to produce sugars, so plants take carbon dioxide out of the atmosphere. Both animals and plants get their energy by respiration, a chemical reaction which releases carbon dioxide into the atmosphere. Carbon dioxide dissolves in the ocean, but this is a two-way process. When the sea gets warmer, some of the carbon dioxide comes out of solution and passes back into the atmosphere. Natural processes like these balance out, so that the tiny percentage of carbon dioxide in the air is maintained. But human activity is now increasing carbon dioxide levels.
So the carbon cycle of respiration and photosynthesis among animals and plants maintains a constant level of carbon dioxide in the air. This keeps the Earth's atmosphere in a constant state suitable for human, plant and animal life. There's more about cycles in nature in the higher tier science programme. That's the end of the section on living things in their environment. That's the end of the Foundation Biology programme. There are other programmes about chemistry and physics, which will all help towards a grade C. There's also a higher tier science programme for those who want to aim higher. And don't forget, there are also the GCSE Bitesize books and the GCSE Bitesize website at www.bbc.co.uk/revision, where you'll find an interactive online science test so you can quickly check what you know.
We were given a choice, slice of bacon or slice of life.
We're getting sunburned.
We chose life, and the rest is history.
I'll hop, and I'll pop, and I'll blow your house down!
It was all about piggy power.
The real story of the Tamworth 2. A Pig's Tale, Easter Monday at 6:30 on BBC One.
Wakey wakey, this is BBC Two and a fun start to Thursday now with CBeebies.
Monday, Tuesday, Wednesday, Thursday.